Hello everyone, welcome to today's live broadcast. Reference component analysis of single-cell transcriptomes reveals cellular heterogeneity and colorectal cancer. I am Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Fluidigm. Fluidigm creates and manufactures innovative technologies and life science tools designed to revolutionize biology through a relentless pursuit of scientific truth. Its core technologies are based on microfluidics and mass cytometry and enable the exploration and analysis of single cells as well as industrial application of genomics. For more information, please visit fluidigm.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as time allows. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located at the bottom left corner of your webpage and follow the process of obtaining your credits. I would like to now introduce today's speaker, Dr. Shyam Prabhakar. Dr. Prabhakar obtained a B.Tech in Electronics and Communications Engineering from IIT Madras and a PhD in Applied Physics from Stanford University. He is the sole recipient of the 2001 American Physical Society Thesis Award in Beam Physics. As a postdoctoral fellow under Eddie Rubin at the Lawrence Berkeley National Laboratory, he led seminal studies of human evolution through gene regulatory changes. In 2007, he joined the Genome Institute of Singapore, where his group uses single-cell RNA sequence, cohort scale, chip sequencing, and other high-throughput assays to uncover molecular mechanisms and diagnostic or prognostic markers of human diseases. In parallel, the group develops computational algorithms for deriving biological insights from functional genomics data. I would like to introduce the doctor now for his presentation.
Okay, so specifically what we want to understand in colorectal cancer is intra-tumor heterogeneity. Now, intra-tumor heterogeneity can take many forms. You could have genetic heterogeneity where the DNA sequences are different because of somatic mutations from cell to cell within the same tumor. And that has been relatively well studied, though there's a long way to go, using single-cell genome sequencing, single-cell DNA sequencing. But then there's functional heterogeneity, which, for example, some cells may be more stem-like in the tumor, some cells may be less stem-like, or there could be microenvironment heterogeneity where the stromal cells could have different properties within the same tumor and actually across tumors also. So this functional heterogeneity within tumors has traditionally been addressed using, say, marker-based sorting. So you use cancer stem cell marker, for example, to purify that subpopulation. But one limitation of that is you need to know what you're looking for already. Microenvironment heterogeneity has again been used, has been studied using bulk transcriptomics, where again you either purify populations or you mash up the whole tumor and characterize the functional states or transcriptomic states. Again, I would say there's two limitations of both these approaches, which is that either you're looking at the whole tumor, which is a very heterogeneous pool of cells, and so you're mixing the signals of the different cell types, or you're trying to pull out what you think is a pure cell population, but then you don't know which marker to use, and then you're really biased, so you only find what you're looking for in some cases. So we believed quite a few years ago that the way to go here was single-cell transcriptomics, where it would be unbiased. We would just take all the cells in the tumor and profile one cell at a time, profile their transcriptomes, and then try to figure out, put all of the pieces together bioinformatically. So that's our approach. And single-cell transcriptomics is very promising. There's already been two great papers in this field for solid tumors from Avi Vergev's group. And the idea is that you can do unbiased de novo identification of tumor cell types, right, without any prior assumptions, and thereby understand, in a panoramic way, so to speak, functional heterogeneity in a tumor. Having done that, you can identify new biomarkers potentially, and also stratify patients. Essentially, you have a more powerful microscope to look at the tumor and thereby to look at the patient. And that should lead to better patient stratification than you can get from bulk tumor analysis. I should say also right now, patients are already stratified based on their genetic mutations in the tumors. We really hope that it should be possible, and I'll show you some results there, that they could be stratified based on the single-cell transcriptomes as well. Then you can find new cell type markers and then do the more, shall we say, biased analysis where you already know which cell type you're looking for, but the cell type you're looking for is really discovered using single-cell transcriptomics. And we can also ask basic biological questions about epithelial tumors, for example. Are they undergoing epithelial-mesenchymal transition? And if so, you should be able to see that in the transcriptome. Are we seeing cancer stem cells? Is there just one discrete stem cell population, or is there a continuum of stemness, as some recent studies have shown? And then maybe discover cell types that were never known before at all. So these are like, cryptic species. These are cryptic cell types where they're subtly different, and they have the same normal FACS marker, but they actually they have different functional properties and different phenotypes. Again, we should be able to identify that bioinformatically from all this. So that's why this field is exciting, and that's why we got into it. It's also challenging. It's not straightforward, but methods are improving, I'd say. So one challenge for epithelial tumors is that cells stick together and they don't like to be dissociated. So I'm using the analogy to the Corleone family. Like any good mafia family, they're very close-knit and they stick together. But then when Fredo dissociates from the family, goes off to Vegas, starts behaving weirdly, and then eventually he gets killed. And that's what happens to epithelial cells if you dissociate them. So they change their behavior, and so you really have to be careful how you handle them. Single-cell transcriptomics or any single-cell assay is noisy because you're measuring a smaller number of molecules. So that's something to be aware of. And it's not just noisy. Noise can be settled, or noise can be combated by averaging a lot of data sets. There's also technical bias from cell to cell, and I'll show you what that does to your conclusions in a minute. And finally, it is more expensive than bulk transcriptomics because you need to do many cells. So how do you address all these challenges? Firstly, in our lab, we use a custom dissociation protocol that's relatively gentle and hopefully does not perturb the cells too much or cause them to die too quickly. I'll show you new algorithms we're developing to fight noise and bias. And the cost is continuously dropping as technologies speed up and scale up. So that's very promising. So I'll start with the algorithms we've developed, which are not cancer specific. But really, if you want to get into this field and you want to learn something from single-cell transcriptomic data, I would argue you need algorithms that are custom developed for single-cell analysis. As I'll show, the standard bulk algorithms don't always work because single-cell data look very different from bulk data. So the three algorithms you will need to do this tumor analysis, or perhaps any complex tissue analysis using single-cell transcriptomics, are firstly normalization. You need to normalize the cells against each other so you can compare them. Then, having normalized the data, you need to cluster the cells. I'm showing a cell-cell similarity heat map. You can see these little squares, red squares, are presumably cell types. They are groups of cells that look like each other at the transcriptomic level, and presumably they're the same cell type. But that's again challenging. As I'll show you need an algorithm for that. And then, having identified cell types bioinformatically, you want to be able to differentiate between two different cell types and say which gene is upregulated in the cell type, which gene is downregulated in the cell type, and you need a separate algorithm for that as well. So why a new normalization method for starters, right? There's well-known bulk RNA-seq normalization methods. The main problem is with those methods is that they're designed for data that look like bulk data, and this is what they look like. What I'm showing here is 6 superimposed bulk RNA-seq data sets from 2 time points, and this is the distribution of expression values. Okay, so x-axis is expression, log expression, you can say, and you can see there's very few very few genes at the high end with 10 raised to 5, 10 raised to 6 expression, and most genes are around 10 raised to 2 to 10 raised to 3 expression. And this distribution is really uniform across the 6 sample types, so the distributions land on top of each other. So almost any bulk normalization method will work on data that look like this. It's not too hard. This is what single cell data look like. Here I've superimposed 6 single cell transcriptomes. As you can see here, the expression distributions are very different. And it's not simply a matter of cell cycle variability or genuine biological variability between the cells, because we see this pattern even in post-mitotic cells. So there's something else going on, and so we really need a bulk transcriptomic normalization method will only shift these curves around. It won't make them look like each other. So that is a challenge, and you could get false positives. Another issue is that you don't detect very many genes. So compared to bulk RNA-seq, in bulk RNA-seq you detect 10,000 plus, so 10,000, 15,000 genes in one data set. Single cell RNA-seq gives you only a few thousand genes, and the worst case, sometimes if you have a tumor sample that's been sitting on the in the hospital for a long time before it gets to you, you could detect only 1,000 or 800 genes. So most genes have a 0 detected expression value, and that again confuses conventional methods. So what's our approach? We developed a method called PQ normalization, and we've even built on that further, but today I'll just talk about PQ normalization. It's pseudo-counted quantile normalization, very simple. Starts with classical quantile normalization, which takes these very different looking expression distributions and normalizes them so now they all look the same, right? And that's a transformation you want to effect. There's still a problem. Each cell has a different number of zeros because each cell has a different quality of data. And that's where the pseudocount comes in. We use not an expression pseudocount, but a rank-based pseudocount. And I don't have time to get into why that's useful, but that's really critical to allow you to compare cells across each other. And as I'll show you, this pseudocount really gets you better performance than normal quantile normalization. So here's one way of benchmarking it. And I should say, for all our algorithms, we do benchmarking. We don't just develop the algorithms and run it. We use independent benchmarks before we apply it to tumor data. So here we are benchmarking our single cell normalization by applying it to 6 different single cell RNA-seq data sets. The one in the middle, the mouse brain, we generated ourselves, and the others were downloaded from published papers. Each bar represents the amount of technical bias in your after normalization, right? So you want the technical bias to be very small, and so you want that bar to be very low after normalization. As you can see, the black bar, PQ normalization, gives you the lowest technical bias of all, and the others, you might recognize some of the other normalization methods that are widely used. So that's great. You want to minimize technical variation in your data set. Another benchmark is how good are your differential expression calls. After normalizing, if you normalize well, you'll get accurate differential expression calls. If you normalize badly, your differential expression calls will not be very good. And the way we benchmark that is we took corresponding bulk RNA-seq data, found differentially expressed genes, and said, can we recapitulate those DE genes in the single cell data? And that gives us a way of benchmarking accuracy. So the y-axis here is accuracy. You can see here again the PQ normalization is the most accurate at finding differential expressed genes from single cell data. And here it clearly outperforms quantile normalization, traditional quantile normalization on the Patel data set and the Trapnell data set. And those are data sets where the number of undetected genes is very different between cell type A and cell type B. And that's where you need the rank-based pseudocount. The third way we benchmarked it was we said, let's apply it to a complex data from a complex tissue, mouse brain in this case, because we want to analyze colorectal tumors, and that's quite complex too. So we dissociated cells from mouse brain, and I think we did about 600 single-cell RNA-seqs, perhaps more. Now in this heat map, you see that each column is a cell, and each row is a marker culled from the literature. So you've got astrocyte markers, neural markers, oligodendrocyte markers, and so on. Now from the heat map, you can see that if you know the markers, you can tell the cell types apart very nicely. And we got all the major brain cell types here. But often you don't know the markers in advance. And we want our algorithms to work so well that they can cluster the cells without knowing the markers in advance. So which normalization method gives you the best clustering? And here's what that looks like. So each row here is one normalization method, and the top few rows are all the popular bulk RNA-seq normalization methods. The last row is PQ normalization, and each dot is a cell, and cells of the same type have the same color. So you can see that the most normalization methods do a decent job of helping you cluster cells by type, but you see long streaks in these PCA plots for many normalization methods, and those streaks are essentially technical variation. They're not biological as far as we know. Only PQ normalization, and to some extent Q normalization, but mainly PQ normalization, causes the cells to clump together very cleanly by cell type. And you don't see these long streaks anymore because it's really minimized technical variation. So by three benchmarks, PQ normalization seems to be superior to conventional normalization methods, and that's because it's specifically tailored to single-cell RNA-seq data. So that's the first algorithm you want. Next, you want a clustering algorithm, but I'll talk about the differential expression algorithm first, because that's published together with the PQ normalization in this bioarchive paper. So this is called nodes, and the key thing to remember about nodes is that it's a non-parametric method. And the reason we opted for a non-parametric method was that non-parametric methods make very few assumptions about the data. What we found is that more or less any parametric assumption you make about single-cell RNA-seq data, whether it's log normal or negative binomial or zero inflated negative binomial, ends up being violated by single-cell data to some extent because of technical variability. And actually, non-parametric methods are just as powerful as parametric methods when you have a large number of data sets, which of course is the case. You have a large number of cells in any single cell data set. So there's no downside to using non-parametric methods, and I would argue there's a huge upside in the sense that your statistics, your p-values are not distorted by deviations of the data from your assumptions. So the other advantage of non-parametric methods is that they are relatively quick. You don't need to do parameter estimation or optimization. And so first, I've actually just summarized these bullet points. Let me get to the statistics we use specifically in nodes. We use a combined statistic, which is the Tibshirani D statistic, and also the Wilcoxon test statistic, which is again, and these are two non-parametric statistics for identifying differential genes. What we found is that one of them works well, one of them doesn't, but when you combine them using Fisher's method into one statistic, you do really well and you get very accurate, okay, I shouldn't say very accurate, quite accurate differential expression calls. And I'll show you how that works in a moment. So first, I'll show you the speed of the algorithm. So here the x-axis is the number of cells we processed. Y-axis is how much time it took on a conventional desktop computer. As you can see here, it takes a few minutes. So nodes takes a few minutes, even when you go beyond 1,000 cells. Whereas methods that are devised for bulk RNA-seq, they don't scale very well, right? They can take up to an hour, or they can, or even more. or they run out of memory. And that's a challenge. So, they take a lot of memory and they're slow. That's a speed advantage, and I would say the scalability advantage, which is very important because new technologies are spitting out thousands of single-cell RNA-seq data sets in one run. You want something that's scalable and fast. And then there's performance. So here you can see how accurate the differentially expressed gene calls are by nodes and by a bunch of other D callers, a bunch of other statistics that are widely used. I should say here, to make a fair comparison, we used PQ normalization for all of these DE callers, and then called, and the only difference was that the DE caller was different. Actually, I'm sorry, I think we used a trim mean of M values here for all the methods. So nodes, to summarize, nodes is faster and also gives better performance on all of these benchmark data sets. So finally, you want an algorithm for clustering single-cell transcriptomes, and then you're ready to go. You're ready to analyze solid tumor data. Now, before getting into that, we thought we would first benchmark existing methods. And for that, what we did is we did single-cell RNA-seq on about 7 cell lines. For all 7, we did the same normalization, and then we clustered the cells by various methods. You can see a PCA plot, which is not what we actually use to cluster the cells always, but it gives you an illustration of the performance. So this is conventional hierarchical clustering, the equivalent of that in a PCA plot. Each dot is a cell, each color is a cell line, and the dots cluster by color reasonably well. But you can see here there's a lot of mixing as well at the left edge of this PCA plot, where different cell lines are mixed together. That's not good. Also, you can see here in the black dotted circles, you have 2 batches of the same cell type separating out very clearly in your PCA plot and in your clustering dendrogram eventually. So that's a serious problem. Batch effects, if batch effects in cell lines are going to confound your clustering, then for sure batch effects in your clinical samples are going to confound your clustering. And so that was a serious concern for us. So we said, let's use another popular method, which is to use PCA itself to reduce the dimensionality of the data, project it onto the high loading genes, and then cluster the cells. Again, you see you get reasonable clustering, but there's a lot of mixing of cell types. And again, you see major batch effects. We also use the backspin algorithm, which is a single cell specific clustering algorithm. Again, batch effects and mixing of cell types. We tried the race ID algorithm. Again, same thing. We saw batch effects and mixing of cell types. And we presume that this is because there is profound technical variability even in cell line data, which are of relatively good quality. And so for sure, you'll get this kind of variability in primary tissue samples, which would tend to be of slightly lower quality. Now, below I've shown the clustering dendrogram equivalents for all these clustering methods. And again, each row is a single cell. You can see here that the cells cluster by color, but then there is some mixing, as you can see visually in the PCA plots as well. So we really need a new algorithm for clustering single cells that's relatively resistant. You can't ever weed it out perfectly, but relatively resistant to batch effects and relatively efficient at clustering cells together by type and not mixing them. And so that's what we did. We developed an algorithm called RCA, which stands for Reference Component Analysis. The key advantage of RCA over conventional methods is that RCA uses more information than just the information in your data set itself. RCA also uses information from public databases of bulk transcriptomes and looks at the variability in those bulk transcriptomes across cell types and tissue types and use that to guide the clustering. So it's still an unsupervised clustering, but with external information. And as you can see from the PCA plot as well as the dendrogram now, cells are clustered almost perfectly by cell type, and there's no batch effect. So the cells from 2 different batches are exactly on top of each other in the PCA plot, and perfectly intermingled in the clustering dendrogram. So with these three algorithms in hand for normalization, clustering, differential expression, we had the confidence to go ahead and look at primary tissue samples. Right? So here's a single-cell RNA-seq workflow for solid tumors. We started with resected primary tumors, colorectal cancer, came to us, and we gently dissociated them. As you can see, there's still clumps of tissue after dissociation, and there's debris as well. So all of that needs to be addressed. Then sometimes we FACS sort the cells to purify single cells by their size, shape parameters, and separate live cells from dead cells as well. We don't usually do that right now because what we find is that FACS stresses the cells, and they're already stressed out from ischemia for a long time. And so we didn't want to stress them any further. So generally, we just take the cells either with FACS or without FACS more often, load them onto the C1 chip. So I'm showing here an image of the Fluidigm C1 instrument, which some of you may be familiar with. So all of the data generated here were done using the 96-cell microfluidic chip. And you can see an image of that on the right-hand side. That's one capture chamber on this microfluidic chip. And you see the white dot there in the middle is a single cell captured in this chamber. The big advantage for us of this pipeline is that we can, of this platform I should say, is that we can image each capture chamber and say, is this a single cell? Is this an empty chamber? Is it debris? Is it a doublet? And so on. Take notes and then sort them out later. So we're only looking at single cells. And I should say this is absolutely crucial for us, the imaging part, because we don't want, say, an epithelial cell sticking to a mesenchymal cell and forming a doublet, and then its transcriptome will look like a mixture of the two, and we'll say, aha, we're seeing epithelial-mesenchymal transition, but actually it's just a doublet artifact, or maybe there's debris from a mesenchymal cell sitting in the chamber. You want to know all that, and that's why we use imaging very carefully always on all our tumor samples. And then the cells are lysed in the microfluidic chambers. The RNA is reverse transcribed, then cDNA is preamplified. It's dumped into a 96-well plate, and then you do the library prep offline. And then you got your libraries. You got your 96 from each chip. You got 96 single-cell RNA-seq libraries ready for sequencing. Take the sequencer and then map the reads and goes to our server, our compute server. And then we do all of the bioinformatics that I just described the algorithms for. So that's a whole workflow. Here's our data set. We had, this is our initial data set. We're, of course, scaling up. But what I'll show you today is from 11 patients. So 11 resected primary tumors. And in 7 of those cases, we also had nearby normal tissue, the nearby mucosa, which are a bit distant from the tumor for comparison. And if we added up all of the tumor and normal samples, we had about 1,600 single-cell RNA-seq data sets. We had a very stringent QC. Well, okay, first I should say this is unbiased. We're looking at tumor and normal, and within the tumor and within the normal, we're looking at all of the cell types. We're not purifying any, because we really want to understand the cancer cells and their ecosystem. So as I said, we did very stringent QC on these cells because they've been ischemic for a while, and so you don't want to analyze all of them. Many of these cells are dying. So after QC, we were left with around 600 single-cell RNA-seq data sets, tumor, normal, all of the cell types. And for QC, we used the number of mapped reads and also the percent of mapped reads that are exonic. And those were our filtering criteria. And also, we use the number of detected genes as a filter. So, okay, what can we learn from these cells? Of course, we normalized the data using PQ normalization. And then the first thing we did was clustering, because we've destroyed the cells. We don't know even which cell type it is a priori. We need the bioinformatics to tell us which cell type it is, and we need the bioinformatics to cluster cells by cell type. So you can see here, first we started by clustering cells from the normal tissue. Each row is a cell, each column is an expression signature. You can see here these cells cluster quite nicely. You can visually see the clustering. The cyan cells in the top segment are epithelial cells. Just below that, the red cells are the fibroblasts. The main thing to note here is that the cyan cells, the dedifferentiated stem-like cells, make up the majority of the population, and which is what you would expect if you're looking at a tumor. The cyan cells were actually a minority in the normal tissue, which is again expected. So I should stop here and point out that already you can see here that if you do bulk transcriptomics, and if you do bulk transcriptomics, and you have different cell proportions, then you could get confounding, right? So if your stem-like cells are more abundant, all of their markers will come up in your bulk tumor transcriptome. And you'll think that all of those markers were upregulated, but that's not true. It's only that cell type itself is more abundant. And that's, again, a major reason why we like to do single-cell transcriptomics, so that we don't get confounding from cell type abundance. Okay, so I don't know if you caught the last thing I was saying, but if your cell type proportions change, then you could get artifactual differential expression, and that's a major concern. So then the next question we asked was, are we seeing epithelial-mesenchymal transition? So we decided to dive right into it. The background is that there have been some studies recently saying epithelial-mesenchymal transition is not necessary for metastasis, at least in a couple of mouse models. In fact, the metastatic cells don't seem to have ever undergone epithelial-mesenchymal transition. And secondly, there was also a study showing that it's the fibroblasts, published not too long ago, showing that it's a fibroblast in the colon tumor that express the EMT genes rather than the epithelial cells. So that's a backdrop, and we said, let's look in our own data. So in this heat map, you see here, each row is an EMT marker. By EMT marker, I mean really mesenchymal markers. Twist, Zeb, snail, all these mesenchymal transcription factors that should come on in epithelial cells when they undergo this transition to a mesenchymal state. And presumably, at least the theory is that allows them to detach from each other, because epithelial cells don't like to detach. And then it allows them to migrate to a different site and form a metastatic tumor. So over here, as I said, each column is a cell, and the blue cells, the dark blue bar at the top, indicates epithelial cells. So the upper heat map is normal, lower heat map is colon tumor. You can see here that the epithelial cells don't seem to upregulate EMT markers. And I'll see if I can indicate that here. So this is the zone you're looking at. Here you can see EMT markers are not very well expressed in the normal epithelial cells. They're not very well expressed in the tumor epithelial cells either, right? So they're not upregulated. On the contrary, you see here that it's the fibroblasts in the tumor, the CAFs, the cancer-associated fibroblasts, that are really upregulating these mesenchymal markers relative to normal fibroblasts. And that's consistent with the recent study showing the same conclusion, only here we show it directly using single-cell RNA-seq on a primary tumor. We decided to quantify this observation because, as you can see here, it's visual, but it's not quantitative. So we gave each cell an EMT score, which is basically just we took the average of normalized average expression of the mesenchymal markers for each cell. The blue dots here are the epithelial cells, and here you see that there's normal epithelial cells over here on the left. So I'm just circling the normal epithelial cells, and I'm circling the normal fibroblasts. So you can see here the normal fibroblasts are mesenchymal already, so they have higher EMT scores. And then the tumor epithelial cells, which I can just draw a line through here, have about the same EMT score as normal epithelial cells, but the tumor fibroblasts have statistically significant upregulation of these EMT markers. So this is precisely what the previous study showed as well, that the EMT signal that you get in your bulk tumor transcriptome is actually coming from the fibroblasts. It's not coming from the epithelial cells. We can't rule out, of course, that there's 1 or 2% of epithelial cells that are expressing, that are going through EMT. But we do believe that if a large fraction of epithelial cells were undergoing EMT, we would see it. Now, one could worry, of course, that one could be concerned that these epithelial cells, what we are calling fibroblasts, are actually epithelial cells. They've changed their transcriptomes so completely that we are fooled into thinking they're fibroblasts, right? But the epithelial cells that underwent EMT. So to guard against that, we did exome sequencing on these same tumors, on bulk tumor, identified the somatic variants, and then went back to the single-cell RNA-seq reads from these cells and said, okay, do we detect those same somatic variants if they overlap an exon? Do we detect them in the single-cell RNA-seq data? So what you see in this contingency table, this 2x2 table on the right-hand side, is that of the 5 CAFs we looked at, this is not a large number, but none of the CAFs that we looked at had somatic variants in there. Only the epithelial cells had detectable somatic variants in their RNA-seq reads. So this gives us confidence. I mean, it's not a large statistical sample, but it's still consistent with our annotations being correct of epithelial and fibroblast. And so here I'm just citing the paper that I had referred to before, studies consistent with their work, saying that it's the fibroblasts are giving the EMT signature. This is quite different from in vitro studies of cancer cells where when you culture them in vitro, they do seem to have a genuine mesenchymal phenotype. So perhaps this is a distinction between what's happening in the primary tumor and what's happening in cell culture. We also, one could also worry that this is an artifact of our processing. Once you dissociate the tumors after resection, so on, something different happens and they lose that EMT signature. But we use some markers, and I won't show the data right here, to validate these findings, these EMT findings, in tissue slices using immunofluorescence. And again, we got the same result. The other question we asked was, are we seeing cancer stem cells? And as I mentioned, you could see perhaps a continuum of stemness in the tumor, or you could see bimodal distribution. We found something in between. Okay, so just like we had an EMT score, we have a stemness score, which is a composite of the expression levels of well-known cancer stem cell markers and also some embryonic stem cell markers. So this is the histogram or distribution of stemness scores for red tumor cells and blue normal cells. So we don't see something bimodal, we see something more or less continuous, but we see a tail that I've circled in green of 10 tumor epithelial cells that really have outlier stemness scores compared to the normal epithelial cells. And so this is quite promising. They, of course, express some of the known stemness markers. That's how we identified them. But they also express some new markers, and we're scaling this up to see what those markers are exactly. So then I alluded to the fact that bulk differential expression could be misleading because the cellular proportions are different between tumor and normal. So if you do tumor normal differential expression, what this Venn diagram shows is that a lot of cell type specific genes show up in your show up as bulk differentially expressed genes in large cohorts. And the green circle on the top left is a true single cell tumor normal differentially expressed genes. They also show up in your bulk data, but actually the cell type specific genes, which are to some extent artifacts, also show up. And that's a challenge. I should say also we have not detected a very large number of differentially expressed genes in single cell, and that's because this is just a first pass. We had only a few hundred tumor cells, and we're scaling up, and then we'll have a more complete compendium of tumor-specific, cell type-specific upregulation and downregulation. So again, it's possible. I mean, you can never rule out that even your single cell differential expression is misleading, again, because of dissociation artifacts or something like that. So for all our major conclusions on EMT, stemness, and in this case, what I'm showing is TGF-beta signaling, we like to use tissue sections and visualize them and see that even in the relatively intact tissue, our conclusions are robust. So what I'm showing here is that we had a conclusion from single cell analysis that it was the CAFs, the cancer-associated fibroblasts, that were secreting TGF-beta, and the epithelial cells in the tumor were responding to TGF-beta. And we're looking at this, of course, because TGF-beta signaling is a known risk factor in colorectal cancer and correlated with poor survival. So the rightmost panel you see is on the top is tumor stroma, specifically tumor CAFs, tumor fibroblasts. And the little red dots, I hope you can see them in there, are TGF-beta molecules. You don't see so many little red dots in the top middle panel, and that's because TGF-beta and those are epithelial cells. So TGF-beta is secreted by fibroblast cells and then signals to the epithelial cells, and we're seeing that in this tissue section. So then the next question we asked is, can we actually tell the clinician anything that they did not know beforehand? Specifically, can we stratify patients, right? We don't have a large enough cohort. We have only 11 patients right now. So instead of trying to stratify those 11 patients, we thought we'll use our single cell signatures to stratify patients in public databases of bulk transcriptomes. And here you see the TCGA cohort and another cohort as well on the right-hand side. To cut a long story short, we use our single cell signatures to classify bulk transcriptomes into 3 classes here, the light blue class, the dark blue class, and the maroon class. And then you see the survival plots for patients from these 3 classes. And you can see here that in both cohorts, with the light blue patients had better survival, and this is statistically significant, than the dark blue and maroon cohorts, indicating that our single cell transcriptomes can inform public bulk transcriptome databases. And specifically this light blue cohort, or the cyan cohort, has a lower fibroblast signature, and that is again known to be correlated with good survival. It also has a lower myeloid signature, and there's some debate about whether macrophages are protective or harmful in colorectal cancer. But in this case, clearly our data are showing that the tumors with the lowest myeloid signature, lowest macrophage signature, have better survival. So there are already bulk transcriptome-based classification of patients by survival. So we said, can we improve on that? And what you see here, the 2 columns are 2 bulk transcriptome subtypes, the enterocyte-like subtype and the goblet-like subtype. We found subtypes using our single-cell transcriptomes. We found a dark blue subtype and a light blue subtype. And I told you already the light blue type has better prognosis. And indeed, our bulk transcriptome classes had subclasses with significantly different survival. So we're very excited about expanding on this approach. So to summarize the tumor analysis, we use novel computational algorithms for normalizing, clustering the cells and finding differentially expressed genes. And using these algorithms, we were able to identify cell types in an unbiased way from single-cell RNA-seq data. We can't always take bulk tumor normal differentially expressed genes at face value, though of course they are valuable for corroboration. The tumor epithelium looks more stem-like on average, and then there is a small population, maybe about 4% of our tumor epithelial cells, that look like outliers. They're very high stemness, and we're investigating that further. We don't see transcriptomic evidence of EMT. Actually, the EMT markers are upregulated in fibroblasts in our data and in one previous study as well. This does not mean EMT doesn't happen, but it does suggest that at least in these tumors, it's not widespread. And we validated this using immunofluorescence as well. And lastly, single cell expression signatures can be used to predict patient survival, even when you're starting with bulk transcriptome data and projecting it onto single cell data. So we think there's a lot of promise for this method, simply because it's more high resolution. So I'd like to thank the people who did all the work. So Elise Courtois on the left did all of the experimental work for the tumor analysis, aided by Juliana Tan. Nirmala Ryan did the mouse brain single-cell RNA-seq work, aided by Michelle. Debaka Sengupta created the algorithms for normalization and differential expression. Huipeng Li created the algorithm, the RCA algorithm for clustering, and did all of the data analysis for the colorectal cancer data. And I'd like to thank my collaborators as well. Paul Robson, who was my former colleague, set up the single cell facility here and drove created our single cell tumor pipeline. And Ian Tan is a medical oncologist, specialized in colorectal cancer, working with us on the interpretation, and my funding agencies as well. Thank you.
Well, thank you so very much, Doctor, for that amazing and informative presentation. Hey, before we get started on today's question and session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And with time allowed, we're going to try to answer as many questions as we possibly can. So Doctor, our first question is this. What factors should be considered if interested in analyzing transcripts that are not single-cell copy? Example, line 1, sign 1 elements.
Tumors may well express their own transposons. So it would be interesting to study how they're interacting and perhaps even influencing tumor biology. That said, we have not analyzed lines and signs ourselves. As the question implies, they're all homologous to each other, so it'll be very hard to tell them apart. I would say if you want to do it with long read paired end, so you can start to tell them apart. At least the slightly older lines and signs, you can distinguish one copy from another with long reads. The really recent line 1 insertions, for example, I don't think you can tell them apart. But yeah, I'm afraid I can't address that question too much because we have not done this analysis ourselves.
Oh, thank you so very much. Our next question is going to be, recent publications from Rusty Gage suggest that ploidy is frequent in normal brains. How does this parameter of variable ploidy evaluate in analysis?
Chromosomes to have different copy number. Certainly in cancer cell lines, and fragments can also be variable in solid tumors. There have been some algorithms that can bioinformatically infer structural variation and copy number variation from single-cell RNA-seq data. In our data set, and we were able to reproduce that on public data from Aviv Regev's lab, in our data, we're not able to tell the structural variations apart using the single-cell RNA-seq data. So I would say you want a complementary, ideally, right? You don't want to be doing it. You want to have some DNA sequencing data as well. There are exciting technologies now that can profile DNA and RNA from the same cell. And I think that it will really be essential, or sequencing DNA and RNA from the same cell, that will be really essential for understanding structural variation and how it influences the transcriptome. So in fact, Elise Courtois my postdoc has just published a paper on that. and not DNA and RNA sequencing, and many other people have published that as well.
All right, and thank you. In examining solid tumors, the same concern applies regarding ploidy. If the tumor chromosomal ploidy changes, how does one separate out transcript changes due to this issue rather than changes the transcriptional regulations?
All right, I'll move on to the next question, which I guess is, how do you know the EMT score of tumor fibroblasts is really from fibroblast? That's an excellent question. So as I said, we clustered cells by transcriptome, and the cells that we are calling fibroblasts, they look identical at the marker level, or very similar at the marker level to normal fibroblasts. The ones that we are calling epithelial look very similar at the marker level to normal epithelial cells. Also in the RNA-seq reads, the cells that we're calling epithelial, we see somatic variants in their RNA-seq reads. The cells we're calling fibroblasts, we don't see somatic mutations in their RNA-seq reads, even though there is ample coverage at some loci. There's a lot of reads covering that somatic variant, but we see only the wild type version. So that's our evidence that it's a somatic variant. Sorry, that's a fibroblast and not an epithelial cell.
All right, thank you so very much here. And does the stage 4 CRC tumor have more fibroblast cells than the high EMT scores than the lower stages one?
That's a good question. We do expect to see variability from tumor to tumor. To be frank, I don't remember what result we got. I remember the result we got from looking at cancer stemness heterogeneity. I'm guessing we would have seen some fibroblast heterogeneity as well, but yeah, we would have to look at that. I would say we really need more data. We don't have a whole lot of fibroblast cells from this data set. And actually the dissociation protocol matters as well. We found that when we change the dissociation protocol, subtly we get more fibroblasts, and we're doing that right now to expand our fibroblast data set.
All right, thank you. And there are several approaches to single-cell transcriptome analysis, but most can be categorized as either end or tag counting of full-length analysis. Why do you choose to use full-length analysis, and why do you think gained from taking this approach instead of the end counting method?
Sure. So I think both methods have their advantages. So with end counting and UMIs, or unique molecular identifiers, you can potentially reduce the amount of noise, but of course the noise also depends on the quality of your data. In our case, we liked the full length because we were able to cover somatic variants with the RNA-seq reads. So we could, to some extent, detect somatic variants. And as I mentioned in the case of EMT, that was really important for us. If we have separate DNA sequencing from the same cells, perhaps end counting would not make us lose so much, but still we hope to analyze splicing as well from the full-length single-cell RNA-seq data. It's not, we won't get all of the isoforms because these data sets have a 3' bias, but we will get hopefully the 3' isoforms, or 3' splicing variation.
All right, thank you so very much, Doctor. Do you have any final comments for us today to take away from your presentation?
All right. Close to the end, just a very brief final comment. This was just our first project in solid tumor analysis with single-cell RNA-seq. I think it's extremely promising, at least from our perspective, and we're scaling up in many different directions. We want to look at biomarkers. We want to look at tumor immunology. We're looking at lung cancer as well. We want to see how these tumors respond to drugs and so on. So there's many directions we can take this. And also we want to scale up to have more cells per tumor.
All right. Well, thank you so very much, Doctor, for your wonderful presentation again, and thank you for your time. And thank you to the audience as well for all of your intriguing questions. I would also like to thank our sponsor, Fluidigm, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through January of 2017, and you'll receive an email from LabRoots alerting you when this webcast will be available for replay. We can invite you to forward this announcement to your colleagues who may have missed today's live event. Until next time, goodbye.